So his first principle was called the principle of dominance. That's super easy. He basically just said some traits cover up other traits. The traits that cover up other traits are dominant. The ones that get covered up, he called recessive. You might say, well, what happens when you cross like red flowers and white flowers and you get pink flowers? That's called incomplete dominance, but Mendel didn't see that. There were no traits like that. And had there been traits like that, he would have been really confused because he wouldn't have understood what was going on. So we are going to talk about traits that don't show a clear-cut dominance, but he did not study that. Every trait he studied had dominant or recessive. And as I mentioned, it's one gene, but it had different versions called alleles. And in the case of the one we just did, it was 2 alleles. There was the one for round. I wrote smooth there, but I was using round in the previous slide. And we used big R for that. And then for wrinkles, we used little r to represent that one. So we typically use one letter and then just use capital and lowercase for the 2 alleles. And again, technically you usually see capital letter for the dominant trait, the first letter of the dominant trait, but you don't have to. A lot of times the question they ask you is a multiple choice question anyway, or it's a grid-in where they just want a numerical response. So they're not going to care what letter you use, or even if you use a Punnett Square or how you got your answer. They're just going to be looking for the answer. This is what I mean by alleles. For example, this could be the chromosomes that a plant has for the color of their flowers. This particular plant is big P, little p, meaning they have 2 copies of the gene for flower color. On one chromosome, they have big P. On the other chromosome, they have little p. Their color of their flowers would be purple, if purple is dominant. You could also have a parent that's big P, in which case both of their chromosomes look like this one, or they can be little p, and both of their chromosomes look like that one, in which case their flowers would be white. So today we know what an allele really is it's a code on a chromosome. He didn't know about any of that. He actually didn't even use the word alleles, he called them factors. And he didn't use the word meiosis either. These are just a list of some of his crosses. You don't have to memorize these or anything, but just to give you an example, every single one of these crosses he did had an exact dominant recessive outcome. Inflated versus wrinkled pods. They either came out fat or they came out wrinkled. Nothing came out in between. Even tall and short. In pea plants, they either come out tall or short. So this is how he discovered his principle of dominance. His second principle is called the principle of segregation. The principle of segregation says that during meiosis, again, he did not know the word meiosis, but even though the organism has 2 copies of every gene, technically 2 copies of every chromosome, during meiosis, only one of the 2 copies is going to go to an offspring. And they're just showing a couple of examples of this. But even if you were big A, little a, for a particular trait, you're only going to give to a sperm or an egg either big A or little a each time. The other copy is going to come from the other parent. So even if I'm big A, I can't give my daughter big A. I can just give her one big A. And then the father is going to give whatever the other letter is, either a big A or a little a. So that's the principle of segregation. It just says segregate just means separate. During meiosis, the chromosomes separate, so you only give 1 copy, even though you have 2 copies for everything. You randomly give 1 copy to your offspring. His third principle we already talked about in the previous chapter, and we're going to talk more about this one tomorrow. Principle of independent assortment basically says not only do the chromosomes separate, but when they separate, each pair separates randomly compared to every other pair. So it's completely random if you're carrying big A and big B and little b on 2 sets of chromosomes. You could give big A and big B, but you could also give big A with little A with big B, or little a with little b. In other words, it's completely random which copy of A you give and which copy of B you give. In this particular case, there's 4 possibilities. We can mathematically figure out the number of possibilities using 2 to the n, and that formula is going to come back again tomorrow as well. And I'm going to show you how to apply it in this chapter like we used it last chapter. So that's the principle of independent assortment. So this is supposed to be illustrating independent assortment. It's just the idea that randomly I'm going to give 1 copy of this pair number 1, one copy from this pair, pair number 2. It could be like this, or I give the 2 light blue ones, or it could be like this, or I give the 2 dark blue ones, or I could give the big light blue and the little dark blue, or the big dark blue and the little light blue. Those are all the possibilities. In us, because there's 23 different pairs, it's 2 to the 23rd possibilities. All right, the rest of this vocab is straight from, we've covered most of it already, but it's already written in your notes, so I'm going to run through it really quick. Again, a gene is the code for a trait. An allele is a version of that gene, like round versus wrinkled, like brown eyes versus blue eyes, like curly hair versus straight hair, basically the genes for those particular versions of the trait. Purebred, homozygous, or true breeding are all synonyms. They all mean the same thing. Specifically, if they say homozygous dominant, it's big B, big B. If they say homozygous recessive, it has to be little b, little b. If they just say homozygous, you wouldn't know. Unless they told you homozygous black and they told you black was dominant. Then you'd know it has to be big B, big B. But they'd have to tell you. Heterozygous or hybrid is what we call it when the organism has big B, little b. Technically, if it's a regular dominant recessive trait, the organism is going to show the dominant trait. And these last 2 words I have not used yet, and these are important. Genotype and phenotype. A lot of times the question you're asked, and the ones you're going to be asked today, the last thing we're going to do today, a lot of times the question is going to ask you, what are the genotype possibilities for this particular cross? And they're asking you, typically you give them in either a fraction or a percent. But for example, you might say that for a particular cross, there's a 50% chance of big B, little b, and a 50% chance of little b, little b. That would be a genotype ratio. On the flip side, you could be asked to give a phenotype ratio of a particular cross. So a phenotype ratio would be what they'd actually look like. So if this is black, you would say, okay, so there's a 50% chance, or you could also say 1/2. Fractions just as good, and that the other half or 50% are white. So what they actually look like is the phenotype. A lot of times I tell people to remember think phenotype, physical appearance, genotype, genes. So genotypes the letters, phenotypes what they're actually going to look like. Technically, that's determined not just by the genes but also by the environment. We'll talk more about that actually later at the very end of the chapter. We'll talk more about how the environment can control that. And that brings us to Punnett squares. So I'm going to give you a walkthrough of how to do Punnett squares. This is probably similar to what you learned freshman year, but unfortunately a lot of times, I guess to simplify things, freshman year, immediately they show you to draw 4 boxes. You don't need 4 boxes for every Punnett square. The whole point of a Punnett square is it's a graphic way to represent the possible outcomes of a particular cross. So, for example, let's say I'm crossing big B, with little b, little b. I don't need 4 boxes. This parent, I'm going to represent what are the possible things this parent can give. They can only give a big B. What can this parent give? They can only give a little b. You literally need 1 box. 100% of the offspring are going to come out big B, little b. Will you get the same answer if you do a 4-box Punnett square? Yes, but you're wasting time. Big B, little b, little b, you're going to basically get the same thing I got 4 times. It's not wrong. It's just unnecessary. So to answer me this, how many boxes do you need to represent the possible outcomes of that cross? Yeah, you only need 2. This parent can give big B or little b. This parent can only give little b. That's all they can give. Your answer to the question of what their ratio is going to be is going to be 1/2 big B, little b, 1/2 little b, little b. Could you do it with 4 boxes? Yes. You're going to get the same exact answer and waste time. Big B, little b, big B, little b, little b, little b, little b, little b. Did you not get the same exact answer? 1/2 and 1/2 or 50-50. So the point of a Punnett square, and the reason I show you this, because obviously you're like, well, it doesn't save that much time. Okay, you're right, it doesn't. But if you remember freshman year and you had a teacher that really wanted to challenge you, they might have given you a cross like this. Big B, little b, big C, little c, with little, 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 little. Based on what you learned about Punnett Squares freshman year, you might have been told you need to make 16 boxes to solve this Punnett Square, which is a pain in the butt and does take a lot of time, and it's very likely you'll make a mistake. You don't need 16 boxes. This parent can give big B, big C, big B, little c, little big, little, little. What can the other parent give? That's it. Little b, little c. You only need 4 boxes, not 16. So knowing, and if you don't understand this is a dihybrid cross, don't panic. We're not even going to talk about that today. But my point is, when you get to more complex crosses, understanding that you only need enough boxes to represent the possible outcomes can save you a lot of time. Now, when do you need 4 boxes? Well, you need 4 boxes for this one. So this one says you're crossing big A, little a with big A, little a. You are going to need 4 boxes for this. Because this parent can give big A or little a. This parent can also give big A or little a. And if we do this cross, yeah, you need 4 boxes to represent the possible outcomes. By the way, if you didn't learn Punnett Squares freshman year, you do always put the capital letter first. Like you don't write little a, big A, just so you know. The genotype ratios, if you were asked what are the genotype ratios of this cross, you would say 1/4 big A, 1/2 big A, little a, and 1/4 little a, little a. You could also have said 25%, 50%, 25%. And you could have also said 2/4 instead of 1/2 here. Any of those would work. For phenotype ratios, even though we're using A's, let's just say that big A codes for tall and little a codes for short. What percentage or fraction of those would be tall? I hear half, I hear 3/4, I hear 75, somebody said. It is 3/4 or 75%. If they have at least one copy of big A, they would be tall. So your phenotype would be 3/4 tall or 75%, 1/4 short. So that's how you do Punnett Squares. Yes, you sometimes need 4 boxes, but the point of a Punnett Square is just to represent the possibilities. If you can do the possibilities in your head, you don't have to do a Punnett Square. I recommend it only because if I give you a short answer on a test and you only give me an answer and your answer is wrong, I can't help you. I give you a 0. If you do work and your answer is wrong, but some of your work is right, I can at least give you partial credit. But if it's a multiple choice question on an AP exam or a grid-in where they just want the numerical response, then it's not going to matter how you go about solving it. The faster, the better. No. So you could give me 3/4, 1/4, or 75, 25, and here you could have said 25, 50, 25. Same thing. All right, so to summarize, I have 3 practice problems. These are the 3. If you can solve these 3 problems, then you pretty much have 2/3 of this chapter down already. So